Disease is probably one of the most underrated crop production problems existing in the world. In fact, some specialists maintain that there is greater loss in income from crops because of diseases than because of either weeds or insects. This unit of study deals with crop diseases and disease management. We'll cover the basic characteristics and causes of disease and the principles that underlie the strategies used to manage diseases in crop production systems. The first topic for study is the section on definitions. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. Plant pathology is the study of diseases that affect plants. Plant disease is simply an unhealthy condition of the plant in either form, function, or processes. The condition is caused by a continuous irritation from some biological organism or factor in the environment. The term pathogen refers to the agent causing the diseased condition. Infectious or biotic diseases refer to diseases caused by a pathogenic organism, such as some species of fungi or bacteria. These diseases are infectious because they can be transmitted to healthy plants by the biological or biotic pathogen. Non-infectious or abiotic diseases refer to diseases caused by a non-biological pathogenic factor, such as air pollution or toxic waste. The term pathogenicity refers to the capability of the pathogen to cause disease. Disease organisms vary in pathogenicity and therefore vary in their ability to cause disease. Disease symptoms are those plant characteristics that indicate a diseased condition. Symptoms may be subtle and only detected after the plant appears stunted or after the disease has substantially progressed. Other disease symptoms may show easily detected signs, such as stem rotting or leaf spots. The next topic for study is the section on impact of crop diseases. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. One can mistakenly assume that because some fungi, bacteria, viruses, and nematodes cause disease, all species in these groups are undesirable and harmful. In reality, most microscopic species are non-pathogenic and essential to a healthful environment. Microorganisms are important decomposers of organic matter and help improve soil tilth and nutrient recycling. Some help plant roots absorb nutrients and convert atmospheric nitrogen into nitrogen forms available to legume plants. Also, some of the many species of microorganisms provide biological control of other insect and disease pests. Because of the many valuable roles that microorganisms play in our environment, it's important that disease control practices should target the pathogenic species with minimal injury to the broad spectrum and population of other microorganisms. Crop diseases cause tremendous economic loss to farmers every year. It's been estimated that an average of 100 or more diseases affect each major crop, and some production practices increase the disease threat. For example, growing the same crop every year and planting large areas to genetically similar varieties increases the potential for severe disease. Non-discriminant chemical control and rapid transportation can spread new, more chemically resistant pathogens. The economic loss from these diseases results from the damage they cause. Diseases damage plants in many ways. Seed decay or seedling blight can negatively affect crop plant population and distribution. This may result in lower yield, as well as the compounding of other problems, such as increased weed growth. Leaf blight diseases can damage leaf tissue, which lowers the photosynthetic rate. Some scientists state that if more than 25% of the leaf area is damaged by a disease, an economically important loss in yield may occur. Some diseases may increase plant lodging due to root and stem rot. Other diseases may produce toxins that not only lower yield, but make the grain unfit for feed and food purposes. The next topic for study is the section on plant microorganism relationships. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. Plants and microorganisms often live in intimate association with each other. When the relationship is mutually beneficial to both organisms, the association is called mutualism. An example is the association of legumes and nitrogen-fixing bacteria. The legumes benefit from nitrogen fixed by the bacteria. The bacteria benefit from the nutrients and habitat provided by the legume root. Parasitism refers to an association in which the microorganism lives at the expense of the host organism. The microorganism which benefits from a parasitic relationship is called a parasite. Plant disease often results from the parasitic relationship of the pathogenic organism to the host plant. Depending on species, microorganisms can survive and obtain their nutrients on living tissue, called parasites, or dead and decaying tissue, called saprophytes. These characteristics influence their ability to survive and persist as pathogens. Obligate parasites must have living host plant tissue to live and to complete their life cycle. Some obligate parasites, however, can exist for a period of time without living hosts by surviving in dormant structures such as eggs. Often, obligate parasites are very host-specific and will infect only a specific species or variety. Rusts, smuts, mildew, and nematode diseases are examples of diseases caused by obligate parasites. Facultative organisms are those that survive and live on living or dead and decaying tissue. Facultative saprophytes usually live on living tissues, but can live as saprophytes on dead and decaying tissues if living hosts are not present. Facultative parasites usually live on dead and decaying tissue, but can attack and infest living hosts and cause plant disease. Many species of facultative organisms can cause disease and usually attack a wider range of crop hosts than obligate parasites. Obligate saprophytes live only on dead and decaying tissue. Few cause plant disease and usually enter plant tissues previously damaged by other means. Most of the microorganism species known are classified as obligate saprophytes, including the beneficial microorganisms involved in decomposition. The next topic for study is the section on pathogenic microorganisms and competitive characteristics. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. Common pathogenic microorganisms of plants are grouped into either fungi, bacteria, viruses, or nematodes. Fungal pathogens are multicellular, producing thread-like mycelium which invade the plant and absorb nutrients. Fungi produce spores that can be carried by the wind for hundreds of miles. Thus, fungal diseases can spread rapidly and widely over a crop production area. Bacterial pathogens are single-celled microorganisms that reproduce by simple cell division. This rapid reproductive capability allows bacteria to rapidly colonize and parasitize a plant host. Viral pathogens do not have a cellular structure and reproduce only within living cells. They consist of nucleic acids encased in a protein coat and are too small to be seen with a light microscope. They multiply by inducing the host cell to form more virus. Cells can be quickly parasitized by the rapidly multiplying virus. Pathogenic nematodes are members of the animal kingdom. They are minute worm-like organisms that reproduce by eggs. Nematodes have a stylet or spear at the mouth end used to puncture cells. Most pathogenic nematodes live part of their lives in soil and either enter plant tissue through roots or feed on root surfaces. Pathogenic microorganisms in general have several characteristics that enable them to competitively survive in a crop production system. They have evolved complex mechanisms to parasitize and exploit plant hosts. They have extremely capable reproductive systems. In some cases, such as bacteria, populations can double in a few minutes. Generation times are short. Therefore, pathogenic microorganisms have excellent ability to quickly develop new strains and races, which are adapted to new conditions, hosts, and even modern chemical control agents. Pathogenic microorganisms also have excellent ability to survive and disperse in the environment. Facultative organisms can survive on living or decaying tissues, allowing the organisms to reinfect crops in later years. Small reproductive structures, such as nematode eggs, virus particles, and fungal spores, may be transported by air, water, insects, larger animals, or machinery. Reproductive structures may lie dormant in soil or crop debris for 1 or more years, only to initiate pathogen growth when susceptible crop hosts reappear. The next topic for study is the section on infectious disease development and management. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. The potential for crop disease in a given situation is dependent on the host's susceptibility, the presence and amount of the pathogen, and the environment. When the host, pathogen, and environmental factors are favorable for disease development, rapid increases in diseases are observed. Conversely, controlling diseases by altering one of these 3 factors forms the basis for disease management strategies. For example, breeding and selecting crop-resistant varieties are attempts to alter the susceptibility of the host. Some disease organisms can survive on alternate host plants, such as weeds, in the absence of a susceptible crop. Therefore, crop producers may try to remove any alternate hosts in the area to break up the disease cycle. Proper crop fertility practices improve crop growth and healthfulness and enable plants to overcome a certain level of disease injury. Biological and chemical control, plant and soil quarantines, sanitary procedures, and crop rotations are all attempts to reduce the amount or presence of the pathogen. Adjusting planting dates and improving soil drainage are attempts to alter the environment that may be conducive to disease development. For example, many seed and seedling diseases are worse in cool, wet soils. Later planting dates under warmer soils, adequate soil drainage, and proper irrigation management are all methods to reduce seed and seedling losses to disease. The remaining part of this unit is the section on disease identification and control principles. You should learn the identification and symptoms of the disease, the susceptible crops, type of pathogen and its survival, infection and dissemination characteristics, and possible control strategies. Please check with your instructor for the diseases which you are expected to know. You should now stop the VCR and study this section in your text. Then return to the VCR for further tutoring. The first category of diseases is common maize diseases. Stalk rot diseases are caused by facultative fungal pathogens that attack the stems of maize. There are several fungi that cause stalk rot, and some of these fungal species also cause ear and kernel rot. Early disease symptoms are dark brown lesions near stem nodes shortly after pollination time in midsummer. Later in the season, the interior stalk is decayed, sometimes showing pink discoloration. Weakened stalks may lodge during the fall. The stalk rot organisms overwinter in crop residues and produce spores that spread by wind to growing maize plants. Control strategies include the use of resistant varieties, crop rotation, proper fertility to improve stalk strength, and minimizing stalk injury from insect pests. Ear and kernel rots are also caused by facultative fungal pathogens, some of which can cause stalk rot. Two examples of the ear and kernel rots are Gibberella and Fusarium. Fusarium kernel rot causes kernels to have a pink discoloration. Gibberella ear rot often results in the husks adhering or sticking to the grain. Both of these ear rots occur in situations where stalk rots occur. Resistant hybrids, preventing insect damage, and proper harvesting procedures can help minimize disease loss. Drying maize to proper moisture content can prevent further losses to grain during storage. Two diseases that damage the leaves of maize are northern corn leaf blight and southern corn leaf blight. Both are caused by facultative fungal pathogens. Symptoms of the northern corn leaf blight are large greenish tan lesions on the leaf, up to about 15 centimeters long and 3 centimeters wide. The fungus may infect the outer surface of ear husks, but does not usually penetrate through the husks to the kernels. Southern corn leaf blight lesions are usually tan in color and smaller, frequently 1 centimeter long and 1/2 centimeter wide. Lesions on the husks are usually grayish to black in color and may penetrate and extend into the kernels. The fungal pathogens overwinter in crop residue and produce spores in the spring, which are carried by wind to young maize plants. Resistant hybrids and fungicides can be used to control these diseases. The use of pathogen-free seed, crop rotation, and proper planting helps minimize disease losses. Corn smut is caused by a fungus that is an obligate parasite. Infected plants may have smut galls on the ears, tassels, stalks, or leaves. Young galls are whitish and later become filled with a mass of black spores. When the mature gall ruptures, spores are released, which can spread to growing plants or fall to the ground and overwinter in soils and crop residue. In the spring, the spores germinate and the fungal mycelium penetrates the maize plant. Most of the maize hybrids are relatively resistant to corn smut. Crop rotation and avoiding plant damage due to blowing soil, cultivation, or other mechanical injury helps reduce disease loss. Maize dwarf mosaic disease is caused by a virus, which is an obligate parasite. Young leaves have alternating yellow and green stripes. Older leaves often have yellowish-red streaks. Plants are stunted, and ears are small or absent. The maize dwarf mosaic virus overwinters in alternate hosts, such as Johnson grass or the foxtail grasses. Therefore, maize dwarf mosaic symptoms on corn are often more prevalent in fields that have weed problems. Some hybrids are more resistant than others. Crop rotations and the control of grassy weeds can help reduce disease loss. The next category of diseases is common soybean diseases. Pythium seed and seedling rot is widely distributed throughout the world and attacks all types of crops. The disease most often attacks seeds and seedlings of establishing crops and is especially troublesome in cool, wet weather following planting. The disease is caused by fungus in the Pythium genus and is a facultative organism. Symptoms include decaying seeds in the soil and brown, rotting roots of seedlings. Seedling death occurs, which is also referred to as damping off disease. The fungus can overwinter in soil and crop residue. There are few disease-resistant varieties. Providing good soil drainage, planting in warmer, drier soils, and treating seeds with a fungicide helps reduce this disease. Another disease of soybean, other legumes, and vegetables is Phytophthora root rot. The pathogen is a facultative fungus in the Phytophthora genus. Brown discoloration of the stem and lower branches usually occur with Phytophthora root rot. The brownish lesion usually extends above the soil surface, sometimes several inches above the soil line. The fungus survives in the soil and crop residue and is most active in low, poorly drained soils. Crop rotations, improved soil drainage, planting in warmer and drier soils, and using disease-resistant varieties help control this disease. However, new races of fungal pathogens are constantly developing and causing continuing problems. Brown stem rot is another disease that damages the stems of soybeans. It's caused by a fungus that is a facultative organism. The symptoms of brown stem rot are usually not apparent until late in the growing season. Infected stems have a brownish discoloration in the center of the stem and near the nodes, which is visible when the stem is sliced in half. Diseased plants prematurely die several weeks before maturity. The fungus overwinters in crop residue in the soil and enters plants through the roots and lower stems. Control may require a crop rotation in which soybeans, alfalfa, or red clover are not grown in the field more than once every 3 or 4 years. Two similar leaf diseases of soybeans that destroy the photosynthetic area are bacterial blight and bacterial pustule. Both are caused by bacterial pathogens that are facultative organisms. The leaf lesions of bacterial blight are usually angular in shape, following the outline of the small veins in the leaves. The leaf lesions in bacterial pustule are often almost circular. Pustules are formed from the swelling in the center of young lesions. Bacterial blight usually occurs earlier in the season, while bacterial pustule usually occurs later in the season, under warmer temperatures. The bacterial pathogens can overwinter on seed or infected crop residue. They spread to plants by splashing raindrops, leaf contact, and cultivation. The use of disease-free seed and crop rotation helps reduce these diseases. Avoiding cultivation when foliage is wet is also helpful. Resistant varieties are also available for bacterial pustule. Purple stain is a disease of soybeans that causes seed damage. It's caused by a fungus that is a facultative organism. The symptom is a purple discoloration of the seed coat. Sometimes only the hilum is stained purple. Seed yield is not usually lowered by this disease, but infected seed lowers product quality. The fungus overwinters in seeds and crop residues. A primary control method of purple stain is to plant non-infected seed. However, if weather conditions are favorable for the development of the fungus, some purple stain is commonly found in most fields. Another leaf disease is soybean mosaic. The pathogen is a virus and is an obligate parasite. The infected leaf may appear leathery, distorted, puckered, and much thicker than normal leaves. Plants can be stunted, and yields can be reduced by 25% or more. The virus is seed-borne and can survive on the seed for 2 or more years. It can also survive on alternate hosts of some weeds and can be spread by many different aphid species. Planting disease-free seed and using good weed control methods helps reduce this disease. The next category of diseases is common diseases of small grains. 2 common and destructive diseases of small grains are stem rust and leaf rust. Both diseases are caused by fungal pathogens that are obligate parasites. The symptoms of these diseases are similar. Stem rust causes narrow, elongated, reddish-brown pustules on the stems and leaves. The pustules turn black as plants mature. The pustules run parallel with the veins and rupture the leaf epidermis. Leaf rust causes more circular, orangish-yellow pustules that are predominantly located on the leaf blade. Pustules frequently appear to be raised above the leaf epidermis and are more easily identified as individual pustules. Both of these diseases can result in lower yields and chaffy, shriveled grain. The fungal pathogens causing rust diseases have a complicated life cycle. The fungal stage found on growing plants in the summertime is called the uridial stage. This stage is also called the red or summer stage and is characterized by the reddish-brown pustules containing red uridospores. The uridial stage is the damaging stage of the pathogen. As plants approach maturity, the uridial fungal stage is replaced by the teleal stage, also called the black or winter stage. The pustules are black and contain black teleospores. The fungus overwinters as black teleospores on straw stubble and wild grasses. In the spring, the teleospores germinate and produce sporidia, which infect the alternate host. For stem rust, the alternate host is the barberry bush. For leaf rust, the alternate host is the buckthorn bush. Esiospores are produced sexually on the alternate host and are blown to reinfect the small grain to begin a new cycle. This life cycle emphasizes the importance of alternate hosts of disease organisms. In the case of the rust organisms, the alternate hosts are necessary in order for sexual reproduction to take place. In the warmer climates, where freezing weather does not occur, infection by the red Uridea spores may occur throughout the year. Most of the infection from the rust organisms in the upper Midwestern United States comes from spores that are blown in from Mexico and the southern United States. Management strategies used to help control rust diseases include the use of resistant and early maturing varieties, planting early, and removing alternate host plants in the production area. Fungicides are available, but chemically intensive use is required and is not generally economical. Two common smut diseases of small grains are loose smut and covered smut. Both are caused by fungal pathogens that are obligate parasites. In loose smut, a thin white membrane surrounds the smut mass, which was formed where the grain should have developed. Later, the membrane ruptures and black spores are released and spread by wind to other plants. The floral bracts, as well as the kernels, are destroyed. In covered smut, the spores are formed within the pericarp of the Caryopsis and are spread when the pericarp is ruptured by harvesting or other means. The floral bracts are usually not destroyed and remain visible on the inflorescence. The fungal spores can overwinter on seed or in smut balls or smut masses to infect a new crop the following spring. Resistant or partially resistant varieties are available. Treatment of the seed with a fungicide may also reduce the amount of smut infection. Ergot, a common disease of grasses and especially rye, is caused by a fungus that is an obligate parasite. A mass of fungal growth, known as sclerotia, replaced the grain kernel of the infected grass plants. Sclerotia are hard, horn-shaped, purplish-black bodies about 0.2 to 5 centimeters long. Ergot infection can lower yield and grain quality. The fungal sclerotia is poisonous to humans and animals, and its presence in grain diminishes the usefulness for food and feed. The fungus overwinters in the soil or in stored grain as sclerotia, and spores infect the new crop in the spring. Practices such as using ergot-free seed, crop rotation, and mowing or grazing nearby grasses before they flower help control this disease. Barley yellow dwarf occurs throughout the world and attacks a wide variety of grass plants, such as lawn and forage grasses and small grains. The pathogen is a virus and is an obligate parasite. Infected plants have yellowish, reddish, or purple areas along the margins, tips, and blades of older leaves. Plants may be stunted and fail to produce inflorescences and seeds. The virus overwinters in perennial grasses and in the bodies of aphids. It is transmitted to other plants by the movement and migration of aphid vectors. Disease control is difficult. Limited success has been obtained by controlling aphids with insecticides and planting resistant varieties. Newer, more resistant varieties are being developed. The next category of diseases is common diseases of forages. Bacterial wilt is a common disease of alfalfa that affects the vascular system of the plant. The pathogen is a bacteria and is a facultative organism. Alfalfa plants infected with bacterial wilt often appear stunted and weak. Leaves are small, yellowish, and often curled. A cross-section of diseased stems will often show a yellow to brown ring under the surface and discoloration in the central portion during the latter stages of the disease. Bacteria overwinter in the roots and crown of the plant and reinfect new growth. The disease often kills the plant after 2 years, and stand loss steadily increases. Control methods include the use of resistant varieties, crop rotation, and providing good surface drainage in a field. Scald is a disease that affects orchard grass. It's caused by a fungal pathogen that is a facultative organism. Symptoms appear on the leaves as lesions that are oval to elongate, 1/2 to 2 1/2 centimeters long. The lesions will be light tan to whitish in color and are often surrounded by a tan to reddish border. Leaves may be weakened and broken over. The fungus overwinters in old crop residue, and spores infect new leaves in the spring. There are some varieties of orchard grass which are resistant to scald. Providing good soil fertility and good surface drainage also helps reduce disease loss. Brown spot in smooth brome grass is another example of a disease of grasses caused by a fungus in the Helminthiosporium genus. The fungus is a facultative organism. Early symptoms are small, dark brown, oblong spots on the leaves. Later, the spots become more elongated and may be dark purple to brownish in color, and may have a yellow halo around them. A lot of the smaller lesions may coalesce and cause large yellowed areas on the leaves of infected plants. The fungus overwinters on old leaves and crop residue. Brown spot develops fastest during cool, wet weather. Resistant varieties, balanced soil fertility, and good surface drainage of the field help reduce disease loss. The last category of diseases is nematode diseases. Nematode diseases are prevalent throughout the world. The pathogen is the minute, worm-like nematode, which is an obligate parasite. Ectoparasitic nematodes feed on external root surfaces, such as the sting and dagger nematodes. Endoparasitic nematodes enter the host and feed internally, such as the root knot, cyst, and lesion nematodes. The symptoms of nematode disease depend on the nematode species. Infected roots may be stunted or have knots, cysts, galls, or lesions. Diseased plants show symptoms of stunting, nutrient deficiency, leaf yellowing, wilting, and poor yields. Nematodes live all or part of their life cycle in soil. They overwinter as eggs, which can remain dormant in soil for many years. Eggs hatch into larvae, which infect roots of growing crops. Nematodes can spread by rainfall, irrigation, flooding, siltation, and movement of produce, animals, and machinery. Variable levels of nematode-resistant varieties are available. Long-term crop rotations using non-host crops, sanitation and sterilization practices, and chemical nematicides have been used to reduce disease loss. The diseases presented in this unit provide some examples of the many different kinds of diseases that affect crops. Diseases vary among crops, fields, localities, and regions, but the principles of disease and disease management are similar. Remember, a sound disease management program begins with proper identification and understanding of the disease and its pathogen. Please check with your instructor for the diseases which you're expected to know. This is the end of the chapter on crop disease management. You should now be ready to try the self-evaluation test at the end of the chapter in your text.